So today we're going to focus on what goes on in the life of a somatic cell. We use that term in the first lesson too. Somatic cells are all of your body cells really except for the gametes. So we're talking about any cell in the other parts of your body. And we're also going to talk about mitosis, which you might remember hearing about briefly in junior high science in grade 9, but we're going to go into some specifics on it. So this diagram is in a little handout that I gave you, and it represents the cell cycle. Now, like I said before, we're just talking about somatic cells, so the way it works to produce a sperm or an egg cell will be different than this. The reason that we have a cell cycle at all really is twofold. Cells reach a maximum size. Maybe you remember in Science 10 learning about surface to volume ratios and how that was important for the survival of a cell. But there's a point where cells can't get any bigger and still function. So if an organism in total wants to get bigger, it can't just keep making its cells bigger, it has to make more cells. So this cell cycle is going to show us how we go about getting more cells. There's a point where cells get to their maximum and they have to divide. They have to make more copies of themselves so that the organism can continue to grow. Now, if we consider the cell cycle, we could split it into two basic parts. The first part is interphase, and if you look at the picture, you can see that interphase takes up most of the circle. And you would think this means it's the most important part. Probably not. It's just the one that the cell is in most of the time. So when I say interphase about 90%, I'm talking about time, not about necessarily importance. Now, interphase could be subdivided into three different sections. The first phase of interphase is G1. Now, scientists have a habit of naming things and then realizing that the name is dumb and switching the name. So originally, G1 was called gap 1 because we didn't think anything happened there. Then technology advances, we observe more and realize, oh, the cell is actually doing something here. So it got renamed to growth instead of gap. Growth is a better name because that is what's happening. During this part of the cell's life, it is growing, physically getting bigger. One of the things a cell does to get bigger is synthesize proteins. Now, I put a number of hours there, not because I want you to memorize 11 hours, but I want you to get a feel for, if we were talking about a whole day of time, how much of that time would be this G1 phase. So the cell grows, makes some protein, gets bigger. Then the S phase happens. If I had to pick which phase of interphase gets asked about the most often, I would say this one. The S phase is the synthesis phase. And the reason it's called that is because we are synthesizing DNA or making DNA. It's when DNA replication happens. This is important now, but will be important again in the next chapter when we talk about the actual mechanics of DNA replication. Now, this phase isn't quite as long as the first one, only 7 hours comparatively, but still a pretty decent amount of time. When DNA replicates, that is when we make a sister chromatid attached to the original chromosome by a centromere. So if I were to just depict what the cell is like, here's a cell. I'm just going to draw one chromosome with a centromere in the middle. During the G phase, the cell just gets bigger. During the S phase, if there was a chromosome in the cell, it gets a sister chromatid attached by a centromere. So we start out with just the chromosome, cell gets bigger, then we make a copy of however many chromosomes there are. I'm just drawing one here for time's sake. I'm not ever going to draw all 46 chromosomes in these pictures because it would take forever. Then we move on to the G2 phase, which is another growth phase. If you look, the first couple of things are the same as what happens in G1. G1 and G2 are really similar. The cell grows, the cell makes proteins, but pretty specifically here, the cell is refilling its energy reserves because it's going to need it if it's going to go to the next part of the cell cycle. Now, if we take the three parts of interphase together, G1, S, and G2, and we add up those hours, it makes about 22 hours. So out of a 24-hour day, it's around 90%. None of these are firm, written in stone numbers. I'm just trying to give you an idea of how much time is passing. Now, even though I drew some specific chromosomes there so that you could see what was happening during replication, that's not really what the cell looks like during interphase. During interphase, DNA isn't condensed. You can't see chromosomes perfectly formed like that. So during interphase, this picture, which is also in the handout I gave you, is more representative of what the cell looks like. Now, every artist will draw things a little bit differently, and you'll notice as you work through questions, depending on who drew the picture, things look a little bit different, but there are a couple of key things that should be in the picture that will allow you to identify it. First, chromosomes are not condensed during interphase. So you should just see strands of chromatin. So big jumbly mess inside the nucleus where you can't distinguish where one chromosome starts and the other one stops. When we look at actual cells, when we'll be looking at pictures of actual cells tomorrow, it just looks like a dark blob. Like you really can't distinguish individual strands or individual chromosomes. The other thing that's true in interphase is that this, the nuclear membrane, is visible. So you can see it, it's there. And you might think that that's a silly thing to make note of, but it's because in some of the other phases of the cell cycle, it isn't. And that's a thing that you could use to differentiate between them. Now, even though that is 90% of the time for a cell, it's only this first little chunk of the lesson, and we'll actually go into quite a bit more detail about the other 2 hours of the day. So before we do, does anyone want to ask anything about interphase? So the other part of the cell cycle, same picture as before, is cell division. So cell division is this section. It's when mitosis is happening. And if we remember how much time had passed in interphase, 22 out of the 24 hours of the day had passed, so there's really only a couple of hours left over for this phase. So time-wise, it's not taking up most of the cell's life, but in terms of what's the most important piece of the cell cycle, it's this one. This is where all the good stuff happens. This is where we make a new cell. There are 3 reasons that a cell would want to go through mitosis. One of them, like I mentioned before, is growth. If an organism wants to get bigger, they need more cells. But it's not the only reason. There are other reasons that your cells would want to make new cells. The second one is maintenance. And I'm going to use blood cells as an example here. You probably remember in Bio20 learning about red blood cells. Maybe you even remember that they have a finite lifetime. They don't last forever. They last for a few months, and then you need new ones. That's an example of maintenance. Maintenance means you didn't really damage anything, but the cell just doesn't last that long, and you need to make a new one and recycle the old one. Repair would be different. Repair would be you've injured yourself, like you broke your leg, and now you need some new bone cells to replace the ones that were damaged when you broke your leg. Now, the next thing that's true is that not all cells divide. So most cells do, not indefinitely, but they do. But there are some cells that really don't, like nervous cells. Cells in your nervous system aren't set up to go through this cycle the way other cells are. We're learning more about the nervous system every day. It's probably the system that we understand the least well, even though it's the coolest one. The second thing that's true is that a cell isn't going to divide unless it gets a stimulus. So a stimulus would be like a signal, a message, but something has to tell the cell, hey, you should divide. We need more cells of your type. A good example of a stimulus that would tell a cell it's time to divide would be a hormone. For example, GH, which stands for growth hormone. Most of you are probably still producing and releasing growth hormone in pretty high quantities. Probably guys more so than gals. A lot of the gals maybe have stopped growing taller at this point. But growth hormone is an example of something that would tell your body, hey, we got to do cell division. We're still growing. We need more cells. The other thing that's true is this: we age, and I put it in air quotes because you're aging all the time. When I say we age, I mean the visible signs of aging, all of the things that oil of Olay is trying to help people combat, like wrinkles and skin that's not elastic anymore. But you start to see some of the physical signs of aging when our cells can't divide, when they can't replace the ones that are damaged, when they can't maintain the cells that are there. You have something called a telomere at the end of each chromosome. So it's this little repeating segment at the end of a chromosome, and every time the cell divides, the telomere gets a little bit shorter. Eventually, it gets so short that it's not there anymore and your cell can no longer divide. Some cells have an enzyme. Now you might remember that enzymes often end with ASE, so even if you didn't know this was an enzyme, you might be able to guess that telomerase is an enzyme. It is an enzyme that's present, for example, in sperm germinal cells, so the cells that are going to produce sperm, that repairs the telomeres all the time, so cell division can continue infinitely. Technically, a male could produce sperm until the moment that he dies. There is no end necessarily to that process. Not the same for a female producing eggs. There is a limit. So this is some general information about cell division, why it happens, when it happens. We're then going to go through each of the sections of cell division so you can see what the cell would look like and what the features are. So we're on to the first phase of mitosis. Now, I'm a big fan of using memory tricks to help yourself remember things. Very frequently, people will refer to mitosis with the acronym PMAT because that is the first letter of each of the 4 phases of mitosis. Kind of like how you use, which one's English, PEDMAS or BEDMAS? I forget which one's English and which one's French. One of them is English, one of them is French. But prophase is the first phase of mitosis, so the first phase of the cell actually dividing. I've shown you a representative picture and listed some key features. The first of the key features is that DNA condenses. In other words, chromosomes are visible. So instead of the jumbly mass of chromatin that we saw in interphase, we can distinctly see that there are chromosomes. The second key feature is that the nuclear membrane decomposes. Now this picture is showing it already gone. Some pictures will show you the nuclear membrane as like a dotted line. And it really depends if we're trying to depict the beginning or the end of prophase. If it's the end of prophase, the nuclear membrane is already gone. If it's right at the beginning, you might see it in the process of decomposing. The third thing has to do with centrioles. These are the centrioles. The shape of the centriole is sometimes the artist taking liberties of what it actually looks like, but there should be two of them. During interphase, they were sitting together on one side of the cell, but during prophase, they're clearly moving towards opposite poles. When we say poles, like the North and South Pole, we mean sides. So one of the centrioles is moving to one side of the cell, the other centriole is moving to the other side of the cell. Now these are the features that you will typically see in a particular picture, but just keep in mind every person draws things a little bit differently. Just because you don't see every key feature doesn't mean it's not prophase. Sometimes they'll draw prophase without showing the centrioles at all. Sometimes, like I said, prophase will have a nuclear membrane already gone. Sometimes it'll be in the process of decomposing. Because prophase isn't a discrete moment in time, it's like a little section of time, things are changing as prophase progresses. So that's prophase. Notice a few things about this cell in prophase. There are two long chromosomes, a pink one and a green one, and two shorter chromosomes, a pink one and a green one. If we look at the two long chromosomes, we would call those a homologous pair. They have the same length, same centromere position. Now one's green and one's pink, to try to illustrate that one of them came from the organism's mother and the other came from the organism's father. So they're not identical, but they have the same size, same centromere position, and they would have the same banding pattern. The two short ones are also a homologous pair. Now, if I was going to number these pairs, this would be pair number 1, and this would be pair number 2, because the longer chromosomes come first in the karyotype, so we always number the longest pair, pair number 1. Now, because I have pairs, this cell is diploid, so 2N. And in this particular picture, there are 1, 2, 3, 4. 4 chromosomes. I'm not counting how many chromatids there are because they're attached by the centromere, so I just count them as one thing. I'm counting how many chromosomes there are, and there are 4. Now, does anyone want to ask any questions about that? Next phase is metaphase. In metaphase, there are little fibers. You can see them extending there, that form between the spindles. Sometimes we call them spindle fibers. The spindles originate at each one of the centrioles. Now these fibers are made of microtubules, and I'm highlighting that because the fact that they are made of microtubules is something that comes up in questions. There are ways to stop a cell from making microtubules that would affect cell division. Now the point of these fibers is to facilitate chromosome movement. And you should see in metaphase that there is a fiber attached to each centromere. So on the picture, there are little circles on each chromosome representing where its centromere is. And there is a spindle fiber made out of microtubules attached to each one of them. Now, I didn't mention it because it was part of the last thing, but here are the two centrioles, one at each pole because they were going there during prophase. And probably the easiest to recognize thing in metaphase is how the chromosomes are lined up. Each chromosome, along with its attached sister chromatid, is lined up along the equator, which is in the middle of the cell. Metaphase, middle, they both start with M, makes it easier to remember. Now we're looking at animal cells here, and I know that because they're circular. And this particular set of pictures is depicting the middle as the horizontal middle of the cell. I could turn this picture 90 degrees, and this chromosomes would still be in the middle. So the middle doesn't have to be the horizontal middle. We could flip the cell sideways, and they could be going down the middle vertically, as long as they're lined up in the middle. Now that's metaphase, second step of mitosis. Does anyone want to ask anything about key features of metaphase? Now PMAT, so PMA, A is next. A stands for anaphase. Now in anaphase, things start getting pulled apart. The centromeres divide and the chromatids separate. So they had been lined up along the middle of the cell, but now we're going to take those sister chromatids, identical copies of a chromosome, and pull one copy to one side and the other copy to the other side. The chromosomes will usually look bent in this picture. If you pull something from the middle, usually you cause it to fold or to bend. If you look at the ones on the top, they kind of even look like the letter A as they're getting pulled up, A for anaphase. The way that they get pulled is because spindle fibers shorten. So as the fiber shortens, it causes the thing that it's attached to, the chromatid, to come closer and closer and closer towards the pole. Sometimes, but not always, you'll see that the cell looks elongated, so stretched or pulled. And so if you compare this picture to the one before it, the one before still looked like a circle in metaphase. Here in anaphase, it's starting to look more like an oval, even sometimes starting to look pinched in the middle. Last phase, last official phase of mitosis is telophase. Now what's really happening here is we start to see the beginnings of two different cells. In telophase, you might start to see chromatids unravel, forming chromatin again. This picture isn't showing it, and it's really a toss-up whether we show this or not. At the beginning of telophase, the chromosomes still look like chromosomes, nice and condensed. And as we get to the end of telophase, then they start to unravel, and it starts to look more like chromatin. Something that you should definitely see is a nuclear membrane. Not just one, but two. One on each side of the cell, because I just pulled those identical copies of chromosomes apart in anaphase. So at the top, I have a set of 4 chromosomes. And at the bottom, I also have a set of 4 chromosomes. They don't have the identical copy attached anymore, but I have the same number. Now, something that people find tricky all the time is what's the difference between a chromosome and a chromatid? And I could say nothing. They're the same thing. We're just talking about a structure at different points in time. If we're talking about a strand of DNA attached to an identical copy, we call them chromatids. As soon as we pull them apart, we go back to calling them chromosomes. They didn't change, they're the same thing, but we call them something different depending on whether they're attached or not attached. There should be very clear pinching in animal cells. In plant cells, you wouldn't see distinctive pinching. Now, this is the last step that is necessary to divide the nucleus. We very clearly have a nucleus at the top and a nucleus at the bottom. There are two nuclei. But the cells are still kind of attached in the middle. The last thing that has to happen, which is sometimes grouped together with telophase but is really a distinct thing, is called cytokinesis. We already divided the nucleus. Now we have two nuclei. We still have to divide the rest of the cell. So all of the cytoplasm and the contents of it. Now, something that you might have to distinguish, you certainly will when we do our lab tomorrow about mitosis, is a distinction between what it looks like in an animal cell and in a plant cell. In an animal cell, the mother cell, which will be the one that we started with, pinches to form two daughter cells. Even if you are male, we would call this the mother and the daughter cell. Female pronouns got used, and that's just how it is. In a plant cell, instead of having pinching, there's a cell plate that forms. So that would be a visual distinction that you could make between what it looks like in a plant and an animal cell. So here's the mother cell, and here are the two daughter cells. Now there's one other thing that I just want to mention about a plant cell that's different. And maybe you remember this from Science 10. But one of the pieces that we discussed today is present in an animal cell but not in a plant cell. Does anyone know which one it is? I heard someone say it, like they knew the answer. It's the centriole. So there are no centrioles in a plant cell. Things just work slightly differently. There are some other things that are different, like an animal cell doesn't have a large central vacuole, an animal cell doesn't have chloroplast, but that's not really the thing that we're focusing on here. The centrioles is the one that's different. Now, like I said, cytokinesis is distinct from telophase, although sometimes they get grouped together as the last step where we divide the cell in two. Telophase is specifically dividing the nucleus in two. This is dividing the rest of the cell in two. The time between when one happens and the other happens is pretty short, so sometimes that's why they get grouped together as one step. Anyone want to ask anything about cytokinesis? Last thing that I want to look at in terms of notes today are two real-life things that affect or deal with the cell cycle. I picked these two because if I were a betting person and I was trying to figure out what kind of context would they put on an exam about the cell cycle, it's probably one of these two things. Stem cells maybe are things that you have heard of before but don't know a ton about. The reason a stem cell would have something to do with the cell cycle is because stem cells can divide and then keep dividing and then keep dividing and then keep dividing. Cancer is a condition where something is going wrong with the cell cycle. So what I would like for you to do, rather than just have me tell you everything, is take a look at the 2 pages that are listed in your textbook there.